In this video and the related paper, we perform autonomous environmental sensing missions with a robot and present the data gathered in an experimental campaign. Measuring physical and chemical quantities in water bodies is important to study and understand lake ecosystems. Limnological measurements are typically performed by manually deploying vertical profilers or by installing static sensors. While these can have a high vertical resolution or temporal resolution, since they cannot traverse laterally, the horizontal resolution is limited. On the other hand, robots carrying sensing probes can measure along the trajectory and gather data at high spatial resolutions and greatly complement these traditional sensing methods. We conducted an experimental field campaign in collaboration with limnologists in a lake, which has a distinct biological phenomenon. This lake has a thin stratified layer rich in diverse species of photosynthetic bacteria. This layer has varying depth and thickness. Therefore, any straight-line robot trajectory will not always entirely be within the layer. The bacterial activity can be studied with the help of high-resolution spatial temperature data, and this is of great interest to limnologists. Underwater robots have additional challenges. Since radio communication does not work in water, the robots need to be fully autonomous, and satellite navigation is available only on surface and not when the robots are submerged. In this paper, we used the Hydromia Vertex AUV, a platform developed in our lab, which is equipped with a suite of sensors, including turbidity, chlorophyll, and a high-resolution fast temperature sensor. The Vertex AUV is less than a meter in length and 7 kilograms in weight. It can easily be deployed and retrieved by one person and was designed as a platform for research on distributed cooperative underwater multi-robot systems. This is a sped-up visualization of the robot trajectory from a mission in Lake Cadagno. In this fully pre-planned autonomous mission, we see that the coverage of the bacterial layer was limited. We then implement an adaptive strategy to fully exploit the benefits of sensing with a robot. We use the fact that the bacterial layer has high turbidity to detect the layer and adapt the trajectory in real time to stay within it. However, staying within the layer is not sufficient. Motor vibrations can degrade the quality of the sensitive high-resolution fast temperature measurements. To mitigate this, we implemented the burst mode, where we turn off all the propellers for a short duration and let the AUV drift forward due to inertia and capture spatial measurements. In this mission, the AUV uses turbidity measurements to locate and track the layer. Once within the layer, during the burst segment shown here in pink, the AUV turns off all the motors. It then gathers high-resolution temperature sensor measurements without the motor-induced vibrations.